Welcome back to the channel. In this video, we are going to study the diagram which are based on two topics, pollination and fertilization in the reproduction and flowering plant. Now, you should know what is pollination. Pollination means transfer of pollen grain from anther to stigma. Now, depending upon the source of pollen, pollination can be of three types: autogamy, geitonogamy, and xenogamy. Autogamy means here the pollen grain will land on their stigma of their own flower only. We say such plants are known as autogamous or they are doing self-pollination. Is it clear? So here we are studying it. This is self-pollination. Clear? But self-pollination. Another is cross-pollination. In cross-pollination, now the flower will be landing from pollen grain will be landing from one flower to the another flower, which can be on the same plant or can be on the different plant. If it is on the same plant, means we are using, suppose let us assume this is one plant and here is male flower and here is female flower. The pollen is landing from one flower to another flower, but of the same plant, you will say this is geitonogamy. It will appear as a cross, but actually it is genetically self. And if suppose the flower are born on two different trees, these are suppose one male plant and this is entirely female plant. And it will pass from one flower to another of different plant, then only we will say xenogamy means cross-pollination. So see this diagram, this is the diagram of cross-pollinated flower. Clear? Now, depending upon the plants can be again of produce two types of flower, either closed flowers or open. So this type of flower is clistogamous flower. Clistogamous flower means they are closed and obviously they will produce seed even in the absence of pollinator. Because they are closed, so they will prefer self-pollination. And these are chasmogamous flower means open type of flowers. Now, few plants such as Viola, Oxalis, Commelina, they are producing two types of flowers. Clear? I hope so this diagram is easy for you. Now see next, depending upon the agent. Here this is a diagram where the wind is used. See here, wind is used as a agent to transfer the pollen grain from anther to stigma. An example of wind pollination is generally seen in the grasses. It is mainly seen in the grasses. Is it clear? Now, example for the water pollination. This is a plant of the Vallisneria. You can see this is Vallisneria. And in this is female flower. This is female flower. First of all, labeling, please understand it. Clear? And now this is enlarged diagram of female flower. This is the one which they are showing it. And this is male flower. Male flower. So female flower and female flower here and last diagram. From this diagram, question can be asked, is Vallisneria rooted and submerged? Yes, it is rooted and it is submerged. And if you see, it is having a thread-like leaves. So that is why it is also known as ribbon weed. And here you will find this is a stigma. This labeling is a stigma. And male flowers are coming in the circle of that area where the water is making a rail-like structure. Means water is there. This is water and these are the male flowers present. Okay. And the moment they come in contact with the stigma, pollination will be done. So Vallisneria is showing epihydrophily. Clear? This is the diagram. I hope so this much topic is easy for you. Now see next. This is the example of the insect pollination. In this case, if you see, this is shown by the orchid. An orchid will not give any reward to the pollinator. What it will do, it will turn one of its petal to resemble the female, and the male think it's a female, come for the pseudocopulation, or you can say orchid is using sexual descent mechanism to get itself pollinated. So these are the methods. They are using wind, they are using water, and they are using the biotic agent. Clear? So total agents, if they ask you a question, please see the NCRT. Biotic agent is only one. They are saying biotic agent only one. Please check the NCERT carefully. Agents of pollination, you will find they are giving it, they are used two abiotic agent, that is wind and water, but biotic agent is only one. Clear? Please, biotic agent, please remember it's only one. Now, this one diagram. So first of all, in the first diagram, if you will see, you will find that the pollen grain is germinating on stigma. Here the pollen grain is germinating on stigma, and here you are finding pollen tube is growing through the style. Is it clear? And see here, many pollen grains have landed, but all pollen grains are not able to successfully make the pollen tube to reach to the ovary. Pollen tube growth is inhibited here. If you see, this pollen is not able to germinate. Only one tube is able to reach properly. So this is the pollen tube. And pollen tube is reaching to the ovary, and inside the ovary is present ovule. Ovule has embryo sac. And we have already studied in the previous video that this is anatropous ovule, and in anatropous ovule, this side is the egg apparatus towards the micropyle. So this is egg cell, only one in number, and this is synergid, which are two in number, and together is known as egg apparatus. Clear? These are antipodals, which are three in number. And this is the central cell with two polar nuclei. Polar nuclei are two, but central cell is only one. So this is the labeling of NCERT. Okay. Now, if you see this diagram, please understand this is very, very important diagram of your NCERT. So this is central cell. Okay. This is central cell, and central cell is having two polar nuclei. These are two polar nuclei. Ploidy of polar nuclei is haploid. You will say haploid nucleus. Clear? Now this is egg cell nucleus. This is the nucleus of egg cell. And this is nucleus of synergid cell. Clear? And you will see in the board diagram, please first focus on the diagram based question. If you see here in the egg, the nucleus is facing towards the chaliceal end, and this is vacuole. In the nucleus, in the egg cell, you will find nucleus is facing the chaliceal end and vacuole is towards the micropyle. But in synergid, nucleus is towards the micropyle and vacuole is towards the chaliceal end. So question language can be this, ki in egg and in synergid, the polarity of nucleus is opposite with each other. In case of egg, nucleus is towards the chaliceal end, but in synergid, nucleus is present towards the micropyle end. Clear? And synergid cells are the one which are having filiform apparatus. This is filiform apparatus which will guide the pollen tube to enter inside the embryo sac. And these are the two male gametes. Please focus. These are two male gamete. Clear? And this is the one vegetative nucleus, or you can say this is tube nucleus. Vegetative nucleus or tube nucleus. And if you see carefully after this, analyze this diagram, come here. You will find one male gamete is making entrance through plasmodesmata connection to do the syngamy. Is it clear? Please focus. One male gamete is going from here to do the syngamy with the egg nucleus. Another male gamete is going up from the degenerating synergid to meet to the two polar nuclei. Now, these two polar nuclei, if you please read the NCERT 11th class, you will find that two polar nuclei just before the male gamete fuse, they become as a secondary nucleus. So secondary nucleus, polar nuclei are haploid, but secondary nucleus is diploid. Clear? And these are two male gametes. The male gametes are going and fusing. So these are male gametes. So one male gamete is fusing with the, let me write it here, one male gamete fuse with egg cell to form zygote, which will develop into the embryo. Another male gamete will fuse with two polar nuclei or sometimes they can give you question secondary nucleus. Okay. The polar nuclei are haploid, secondary nucleus is diploid to form primary endosperm nucleus. Zygote ploidy is 2n, primary endosperm nucleus ploidy is 3n. Again, let me highlight one important point. If they give you polar nuclei, you should know polar nuclei ploidy is haploid. But secondary nucleus ploidy is diploid. Clear? So which lead to the double fertilization. One is syngamy. This is syngamy where the two gametes are getting fused to form zygote. Another is triple fusion. This is triple fusion where the three nucleus are getting fused to form primary endosperm nucleus. Another diagram based question they can ask you, how many nucleus fusion is there? Total, if you see here, 1, 2, 3, 4 and 5. 5 nucleus fusion is there. Total to make zygote and endosperm, total 5 nucleus are fusing. Is it clear? So I hope so in this video, this topic might be easy for you up till here. Now see, this is the diagram which I've explained you and very important diagram of NCERT. First, this is very, very important. Please learn the polarity of nucleus in egg and synergid cell is reversed. Very important. Next, see this labeling. This is the one after fertilization is done. This is the, you have to assume this is your micropyle end, okay? Because in the diagram, see carefully, beta, understand it. Theory is written with orthotropous ovule. We are writing entire theory of the NCERT. If you read NCERT theory properly, you will find with orthotropous ovule. That is why we say polar nuclei are present below egg apparatus. So theory is with orthotropous ovule because they have done on the polygonum type of embryo sac, which was having orthotropous ovule. But angiosperms are having majority anatropous ovule. Clear? So here we are again going with the orthotropous ovule. So you know very well, these are degenerating synergid. These are the one which are degenerating synergids. And this is the one zygote. Okay, zygote. The zygote ploidy, you know very well, it is diploid. This is primary endosperm nucleus. And you know primary endosperm nucleus ploidy is triploid. And the central cell, after fertilization, central cell will now known as primary endosperm cell. Before it was known as central cell, but after fertilization it is known as primary endosperm cell, and these are degenerating antipodal cells. So this is the fertilized embryo sac. Clear? Fertilized embryo sac. Now see next. This is one cell zygote. Now we are studying embryo development. So this is zygote. It becomes into two cell stage. This is two cell stage. And then it becomes to the globular embryo. Globular embryo. Clear? And this is suspensor. This is heart-shaped embryo. And finally, if you see, this is mature embryo. These are the stages you have to learn. They can ask you the stages. So zygote will become two cell. Okay. In the theory, you will find it is given pro-embryo also. One is missing in the theory. Okay. In the theory, I'll just write it pro-embryo also. Pro-embryo. Then it becomes to globular embryo. Then it becomes heart-shaped embryo. And it becomes mature embryo. So this is in the NCERT. If you will read, you will find it properly. And these are the two cotyledons because of the dicot, two cotyledons. And this is plumule. Clear? This is plumule. And this part is radical. So this is the one NCERT diagram which I have explained it. I hope so this much video might be helpful for you. In the next video, we'll be covering remaining diagrams of this chapter. Thanks for watching.